In the last module, we focused on the detailed process of germination and discussed the various determinants and requirements for seeds to germinate. In Module 6, our focus will be on seed dormancy, including its various forms and determinants, as well as methods of breaking dormancy in seeds. The most common misconception about dormancy is that it is merely a state of quiescence or rest in the absence of suitable germination conditions. This definition indicates that dormant seeds will germinate when subjected to conditions that are suitable for germination. But this is not always the case. True dormancy is a state in which seeds are prevented from germinating, even under conditions that are favorable for germination. Dormancy is a genetically inherited trait, but just like any other seed traits like morphology and chemistry, dormancy is also subject to the effects of the environment. The degree of dormancy, therefore, is determined by the combination of the genetics of the plant and the effects of the environment. To illustrate how the genetics of the plant and the environment determine dormancy in plants, let's take a look at this palm tree named Methuselah. This palm was grown from 2,000-year-old seeds that were excavated in Israel. The seeds are pictured here. Because the seeds were very old, the tree that came out of it was named Methuselah after the longest living character in the Bible, who lived for 969 years. The seeds were dug up in 1963 to 1965 in the palace of King Herod, which was located in Masada, Israel. Masada is a natural fortress in the Judean desert overlooking the Dead Sea. It served as a refuge of the last survivors of the Jewish people fighting the Roman army in 73 AD. It was unfortunately besieged by the Roman army, and as with any wars, most of the structures in the palace were destroyed. You can see here the ruins of the palace. Now, during the excavations, seeds of date palms were found in ancient jars that were buried deep in the underground chambers. As I said, Masada is located in the Judean desert. So while temperatures may vary, the air will be mostly dry, and you know that water is a primary requirement for seed germination. With a dry environment, the seeds were able to maintain their dormancy. The protective seed coat, the morphological and chemical features of which were determined by the genetics of the plant, ensured the integrity of the sleeping embryo inside, even after 2,000 years. Generally speaking, plants that have been domesticated show less dormancy than those with little or no history of domestication. What do we mean by domesticated plants? These are plants that have been selected for specific traits for thousands of years so that they suit human needs. Examples of domesticated plants are your cereals, including wheat, corn, and rice. There are also your fruit trees, vegetable crops, and grasses that you grow in your lawn. All of this have been tailored to suit our needs for food or even for aesthetics. Because they are domesticated, these plants generally can no longer survive in the wild. We have to tend to them much like you tend to your pets. And that is exactly the reason why domesticated crops have lower levels of dormancy. Because these seeds, through maternal imprinting, know that they will be cared for from germination to maturation. So there is no strong mechanism for self-preservation because the seeds are assured of the perpetuation of their species. In contrast, non-domesticated species are those that grow even without human intervention. These are the wild relatives of our crops and the grasses in the field that we call weeds. Seeds of these species need to ensure the perpetuation of their species. So say for example, we have seeds of these wild plants in a rangeland, and then one day during the early spring, it rains. The seeds are able to sense the availability of water, which is a requirement for germination. But chances are the seeds will still not germinate. Ecological adaptation are inherent to seeds. If a wild plant has been growing in a rangeland for several generations, the seeds are aware that an early spring rain does not mean available water throughout the spring to the summer. If the seeds germinate after that single rain, they are not guaranteed that water will be available until they complete their life cycle. So instead of germinating, they will just remain dormant in the soil. Silverleaf nightshade is one good example of a plant species that we consider wild. In cotton fields, silverleaf nightshade is considered a weed. We spray our fields with herbicides in the early spring to kill these beautiful plants in preparation for planting our crops. This slide shows you the results of a study that looked at the dormancy of silverleaf nightshade seeds collected from different ecologies. These include a rangeland in Littlefield, as well as the Quaker Farm in Texas Tech, also a rangeland in the university, as well as an urban site. This panel shows morphological variations in the seeds of silverleaf nightshade collected from those four locations. If you look closely, the seeds vary not only in color but also in size. The second panel illustrates the water uptake patterns of the seeds. Recall that germinating seeds uptake water in a triphasic manner, the first phase of which is characterized by a rapid influx of water followed by a leveling off. In this panel, we can see that the first phase of water uptake in the seeds occurred within 20 hours, as indicated by this rapid influx of water. This 20 hours was followed by a leveling off, which lasted up to 40 hours. The x-axis here indicates the number of hours. You can see that regardless of the source, all the seeds took 20 hours to complete the first phase of water uptake. In contrast, domesticated species such as cotton and corn requires only 4 to 6 hours to complete the first phase or the imbibition phase. The next panel shows the germination rate of the seeds. Seeds of silverleaf nightshade were germinated in sandy soils under controlled conditions. The x-axis here indicates the number of days of germination, and the y-axis indicates percent germination. Obviously, we are seeing variations in the rate of germination of seeds coming from different sources, with seeds from both rangelands showing very poor germination. These are indicated by the blue and also the green bars here and also here. As I said earlier, these seeds were germinated under controlled conditions. It means they were provided good growth media, light, and water. Despite that, the seeds still showed poor germination, which is indicative of their dormancy. Seeds from the urban site, as indicated by these yellow bars here, had a maximum of 80% germination. But if you look at the x-axis, you will see that seeds from the urban site was able to achieve this level of germination after 49 days in germination media. Most crops would have germinated within 1 to 3 days in the field, but because silverleaf nightshade is a wild plant, it has higher levels of dormancy as seen from this data. There are several physical and physiological mechanisms controlling dormancy in seeds. Depending on various factors, dormancy can be classified as exogenous or endogenous, or primary or secondary. When dormancy is under the control of the different parts of the seed, it can be classified as either exogenous or endogenous. Exogenous dormancy is caused by conditions outside of the seed's embryo. This means dormancy is controlled by the endosperm or the seed coat. An example of exogenous dormancy is when the seed coat has chemical or physical properties that prevent the seed from taking in water or moisture or air that are key requirements for germination. In contrast, endogenous dormancy is caused by chemical changes within the seed's embryo. Dormancy can also be classified depending on its onset during the process of seed development. When dormancy is classified by its onset, then it can be classified as either primary or secondary dormancy. Primary dormancy is the most common form of dormancy observed in seeds after they are shed from the mother plant. This type of dormancy ensures that seeds do not prematurely germinate while still attached to the parent plant. The loss of primary dormancy results in vivipary, or the precocious germination of seeds. This slide illustrates how viviparine seeds without dormancy can germinate while they are still in the mother plant. The first panel is an example of precocious germination in rice. You can clearly see the shoots here and the roots of the new seedlings while the seeds are still intact in the panicle. The second panel shows viviparine barley, and the last panel shows vivipary in corn. Contrast, secondary dormancy is an acquired dormancy in response to environmental conditions after seeds are shed from the parent plant. So say for example you have cotton seeds that are non-dormant, meaning if you subject them to conditions that are favorable for germination, they will be able to germinate. If these seeds are exposed to extremes of temperature, the seeds will develop a secondary form of dormancy that will prevent it from germinating under temperature extremes. The secondary dormancy will allow delays in germination until environmental conditions become favorable for the successful growth of seedlings. A seed can have both primary and secondary dormancy. Summary, dormancy is a state wherein seeds are unable to germinate even under a combination of environmental factors that are normally suitable for seed germination. It is a genetic trait that manifests as physical and physiological mechanisms that inhibit germination. Dormancy can be classified as exogenous or endogenous, and primary or secondary. Primary dormancy ensures that seeds do not prematurely germinate while still attached to the parent plant. Secondary dormancy is an acquired dormancy in response to environmental conditions after seeds are shed from the mother plant.